You're at the cutting edge of RNA therapeutics. I'm John Cooke, professor and chair of the Department of Cardiovascular Medicine and the director of our Center for RNA Therapeutics. We have a wonderful speaker for you today who's going to be introduced by my colleague, Dr. Christopher Brannon, who's also a faculty member in our Center for RNA Therapeutics.
Thanks, John. It's my pleasure to welcome Dr. Gene Yeo, who's a world leader in RNA research, and his work bridges RNA therapeutics and computational experimental biology for therapeutic applications. Gene's Sanford Stem Cell Institute endowed chair and professor of cellular and molecular medicine at UC San Diego, where he also directs the Center for RNA Technologies and Therapeutics and the Sanford Laboratories for Innovative Medicine. He holds a dual bachelor's degree in chemical engineering and economics from the University of Illinois and a PhD in computational neuroscience from MIT, and also an MBA from UCSD's Rady School, just to be safe. And Gene is a serial entrepreneur and advisor, co-founding multiple biotech ventures such as Eclipse Bioinnovations, and serving on scientific advisory boards from the Allen Institute to Roche. Gene's work primarily focuses on RNA binding proteins and their roles in gene expression. He has hundreds of peer-reviewed publications, pioneering technologies and innovations. Gene has transformed our understanding of RNA processing and development, neurodegeneration, and cancer. And today he's going to share some insights into engineering zinc finger proteins for precise RNA targeting. So just to get started, I kind of wanted to ask Gene about the Center for RNA Therapeutics and Technologies that he co-founded and directs, and also the Sanford Labs for Innovations, and sort of what motivated founding those centers, and I guess what roles do you think they're going to play in sort of pushing preclinical and clinical advancement of RNA-based therapeutics?
Yeah, John and Chris, really great to see you both. Thank you for the kind introduction and invitation to present some of our work here. I'm delighted to talk about both of our initiatives. One is the UCSD's Center for RNA Technologies and Therapeutics. That's a relatively new organization that we have set up within UCSD, and the primary mission there is to leverage our RNA ecosystem here. We have 40 to 50 labs across the Mesa that are experts in RNA technologies or biology related to RNA processing, or have a deep interest in applying RNA-based technologies to disease intervention. And so a couple of years ago, the chancellor at UCSD was very generous in providing support to our vision of trying to enhance the RNA activities at UCSD and on the Mesa, and to really draw in from the community to build technologies that can enable next generation therapeutics, or to accelerate the current generation of therapeutics in the space. We are a year and a half old, I guess, and are starting to build interactions across different other RNA centers, as well as across countries. So we've been starting to talk to different countries who have much more, I guess, focused need on trying to develop next generation therapeutics, and are comfortable with the idea that RNA can provide this basis. And then on the other hand, we have the Sanford Laboratories for Innovative Medicines, which is an independent, not-for-profit 501c3 organization that's primarily resourced by a philanthropic gift from Denny Sanford. And Christina Brewery is the president of the institute. And she and I'm the chief scientist of the CSO, and so we got together to lay out the blueprint of what would be a different path. So oftentimes at our research labs funded by NIH, we do fundamental basic research, we do some early tech development, but we will reach a point where NIH funding may not be sufficient, or it may be deemed not innovative enough to fund the maturation of these technologies or potential therapeutics. And so Temperate Labs plays this role where we are sort of bridging the gap or the valley of death, so to speak, between the fundamental research and then working with investors, working with pharma and biotech to license or to partner or to build joint ventures or to build new companies from these inventions, right? And what's also very unique is that the group leaders that we have brought in, many are, I think all of them are actually alumni from my group that have gone to the biotech industry for a bit and have sort of come back with a deeper understanding of drug development and product development. And they don't have to write papers at Temperate Labs. They can if they want to. They don't have to write grants at Temperate Labs. They can if they want to. But really is to accelerate products to a stage where then there can be clear commercial opportunity. And so it's, I think, a very unique place where you have individuals that are not necessarily focusing on academic career or developing fundamental knowledge, but to accelerate fundamental discoveries quickly into something that can get to patients. And so I think in this day and age, it serves a very unique position in being able to do focus research to commercialize ideas, but we are not at Temperate Labs dependent on at this point the NIH funding or having to actually promote people through the ranks in the academic system. So I'll just stop there and happy to take more questions. Yeah.
That's really interesting, Gene. And certainly you've figured it out. You've been able to take your basic science ideas all the way to the clinic, commercialize them. And so I think this is really an interesting program and that you've described because it does fill a gap. Everyone's familiar with this valley of death between the basic science that NIH funds and the product, its commercialization. There is a gap, but maybe people aren't as familiar with the idea that you need to do some product development before industry is even going to be interested. So that's not funded either by NIH. So I think that's what the Sanford lab sounds like. It's filling that gap by taking the basic science, developing it a little bit further to show its commercial promise so that you can get industry interested or do a spin out of that technology. Really interesting. Do you have?
John, and I think sorry, go ahead.
No, please.
A big part of it is de-risking the technology and the proposed product. And I think what's quite unique also is the synergy among the teams that we have put together. Unlike accelerators, traditional accelerators, or venture studio models, where you have different companies in one physical space, where the companies are not tied together thematically, right? Here we are building multiple shots at multiple goals, right, with a very synergistic group. And so there is a lot of efficiencies that are gathered by this idea. You have shared lab meetings, shared progress, shared experiences with thinking about how to trim ideas or kill them quickly. Unlike academic projects where sometimes they keep going without the balance of prioritizing to something that may have a nearer term return. And so I think this is to me, I'm very excited about the opportunity to help build this organization.
Where does the technology come from? Is it all coming from UCSD, or is Sanford? Would they be interested in taking technology from other universities? How does that work?
So the current instantiation of Sanford Labs is to draw technologies or new targets from UCSD, but one can imagine it can do that for any of the neighboring universities, institutes in San Diego, and there is no reason why we cannot match technologies that are missing from our current portfolio, where together with something that we have may create something of even higher value.
I have one more question.
The goal really is to accelerate biomedical innovation. Yeah.
Really interesting. So you told us about Sanford. Tell us a little bit now about your Center for RNA Therapeutics at UCSD. What do you guys do there? Is it more RNA innovation, or do you actually generate products there?
So the focus at the center tend to be more on technologies that enable RNA therapeutics, right? So it's a little bit different, right? So we have, for example, leverage a lot of our expertise in genome-wide assays, functional genomics, drawing links between genotype to phenotype, RNA, CRISPR, siRNA assays to perturb RNA processing at scale. And these are technologies that are moderately to high throughput in terms of scale, and enables us to perturb many things with spatial technologies in there and so on. And so it's really to provide this type of technologies to folks that are interested in developing hypotheses or therapeutics in the RNA space. So for example, if a faculty member comes to us and say they have an interesting gene target and want to study this target using antisense oligos, siRNAs, mRNA, we would then help with that research, right? We could then design, we could screen oligos, or develop ways to help interpret the data. With others, maybe want to come in to explore some of their small molecules if they have a way to modulate mRNA translation. Then we use our ribostem assay to enable that, right? And so this type of technologies we are continuing to grow, and these platforms are enabling sort of catalyzing the community as well as our own lab to get do better science in the RNA space, and some of the better science will lead to new targets that we can be drugged, or maybe the early stages of something that could be a potentially new product, right? And so it's still sitting in the academic realm, but with an eye towards translation. Yeah.
Well, we're looking forward to hearing more about your academic realm and the research you're doing. But before we start, Gene, I want to ask you, is it okay if we ask you questions during the presentation?
Yes.
Okay.
Absolutely. I can't. Yeah, absolutely.
And let me just let those of our audience know that they can do that. They can call in. They can call 37607, text DeBakey to that number, and then just ask your question. Or you can go to polev.com and enter DeBakey, respond to that activity. And we'll get your question here in the studio, and then we'll ask Dr. Yao. So I think we'll go ahead and get started. If you want to share your screen with us, Gene.
Yeah, let me know if you can see it.
Looks perfect.
Looks good. Okay, I'm gonna also get a laser pointer running there. Okay, so thank you for your time. I'm going to tell you a bit about our work in trying to understand a class of proteins within the space of RNA binding proteins. First, I have to give the introduction to remind everyone that RNA is never naked in the cell. This is true for endogenous RNAs, or RNAs that are delivered exogenously. As soon as they're present in the cell, RNAs recruit proteins that bind them, and these are RNA binding proteins that shepherd the RNA throughout its RNA life cycle. That means that the RNA binding proteins, or RBPs, are coding the RNA and bringing it to different places, processing it, editing it, modifying it, affecting translation, and providing a way for the RNA also to be protected, right? And so we have been studying this class of proteins for a long time, and in our estimation from our large-scale studies of about 150 to now 200 RNA binding proteins, where we have mapped their binding sites on RNA substrates, we think that a large fraction of the transcriptome it's just bound by RNA binding protein. So everything at least 250 million coding nucleotides are bound by at least one RBP, and there are many proteins now in the genome that are thought to encode these RNA binding proteins. And so my group studies basically how RNA binding proteins affect RNA regulation. We are very interested in the mechanisms underlying the regulation of RNA processing by RBPs. We identify RBP RNA regulatory networks. We try to assign molecular functions to these known and predicted RBPs. I'll show you a slide on that. And then we try to understand rules underlying how they recognize RNA and how they modulate it. But at the same time, we are trying to identify RBPs as novel targets for disease and therapeutics, neurodegeneration, neurodevelopment, and cancer. And also to try to manipulate these RNA binding proteins. Perhaps you can use them for therapeutic intervention, if not at least as tool compounds. And a very early study combining many studies, one already knows that for all the RBPs that are starting to appear over the last decade, many of them are still relatively uncharacterized and have no known RNA binding domain. And then I'll segue into why we ended up with this particularly interesting class. One way to quickly assign a molecular function to a predicted or maybe a known RBP where we want to assign a new function is to develop these high throughput tethering assays. So we start with a collection of RNA binding proteins ranging from 700 to more than 1,000 open reading frames. We can fuse them to MS2 coat protein, MCP proteins. These MS2 coat proteins bind to MS2 RNA stem loops with high affinity. And so you can then tether these RBPs independent of their RNA binding motif or preferences to RNA tethered reporter assays. And here in this case, for example, we asked if an RBP can affect stability or translation by being in proximity to within the 3' UTR of a reporter assay. And we've done this for many kinds of reporter systems. And so this one that looks at RNA stability and translation, identify 50 or 100 different proteins that have roles in this area. But also we have just recently, as of last year, completed an analysis of which RBPs can affect alternative splicing, exon inclusion, particularly using the same RBP library, but with different MS2 stem loops within different reporters, in this case, a splicing reporter. And we have different papers for alternative polymorylation, circular RNA metabolism, all under review. And then at the same time, we've been interested in trying to understand rules for protein RNA engagement. And so I take the liberty of embarrassing Chris Brandon here, who was first or co-first author of two of these papers when he was a fellow in my group. And here we have been developing algorithms that leverage mass spec data or sort of protein interaction association data to try to understand rules for how candidate RNA binding proteins bind to RNA and identify novel domains within within proteins that we did not anticipate to be RNA binding. And from some of these studies, also with previous studies from Matt Hansen's group, we've all come to the realization that many of these newly identified RNA binding proteins are enriched for DNA binding domains, especially zinc finger domains. And the left is work that Chris had led, and then on the right is from experimentally pulled down mRNA interactome studies that also identified some of this in the original paper. So we've been quite curious about this class of zinc finger domain containing proteins. It turns out that the human proteome contains more than 1,700 zinc finger proteins. And historically, the research community has focused solely on the DNA binding properties of these zinc finger proteins, but we've been very curious about the RNA binding properties. For those who are even more curious, zinc fingers are very small. There are about 30 amino acids, and they require zinc for the interaction or for stability of the protein, the zinc finger, but also interaction with nucleic acids. And so below you can see different types of zinc finger domains. Okay, and then on the right you can see that if you take this class of genes and you just run gene ontology, most the strongest categories are still transcription and chromatin. But recently, work from my group, led by Maya, and in collaboration with many other groups below, as well as a companion paper by Jack Greenblatt's group, the same issue of molecular cell. We have now clarified that actually many zinc finger proteins domain containing proteins bind RNA and affect RNA processing and affect multiple layers of cell biology even within the same cell. So it turns out that there is excitement now because one would have previously annotated all zinc finger containing proteins as DNA or transcription factors or something, but now I think a large fraction of them are RNA interacting proteins. Okay, here's some evidence from one of my previous students who's now a postdoc at Scripps. So Lisa worked with her co-mentor Kevin Corbett and myself to demonstrate that we can actually understand but also change the specificity of at least one of these RNA zinc fingers with rational design. So this is a ZRNBP2B2 zinc finger that has the PDB structure shown on the left that recognizes a simple GGU trimer. And of course, in this protein, you have many zinc fingers that are found in the protein to then bind longer stretches of sequences. But this is just a one of the zinc fingers. And so here we have performed rational design to evaluate if we alter the zinc finger on the protein side, does it change the specificity for the RNA? And Lisa had approached this two ways. So one way, she basically developed a library of single or double mutations of the zinc finger and then applied RNA bind and seq, which is like a high throughput in vitro selects, to ask how each mutation affects the preference for the nucleotide. And she found one of these mutation changes the affinity of the zinc finger from GGU to GGG, and it's only one nucleotide change, but this has not been achieved before, and we were very excited that there is this possibility you could screen and rationally design new preferences for zinc finger domains. She had also completed during the pandemic this molecular dynamic simulation to validate these hydrogen bonding changes when she makes the variant. So on the right here, you see the wild type zinc finger recognizes GGU, and the interaction between the amino acid and the base is about 2.8 angstroms. Now, when you make this single point mutation, the NR mutant loses a critical hydrogen bond with GGU, so it's now 5.1 angstroms in interaction space, but then recovers that bond when you have a G instead. So we can at least understand now how the protein domain interacts with the specific nucleotide. And so this has been continuing to expand in my group. We have now made libraries for hundreds of zinc finger domains, trying to understand how they interact and to perturb these interactions. But as a proof of concept that even wild-type zinc fingers could be helpful in a therapeutic regime, we have focused on this hexanucleotide repeat expansion in the C9orf72 gene in the indication of ALS. So this is what we call C9 ALS. It is the most frequent cause of ALS. Here you can see that it takes up the largest fraction of patients with familial variants in ALS and also the large fraction in sporadic ALS. Now, C9orf72 driven ALS is caused by a large hexanucleotide repeat expansion of GGGCCs that's present in the first intron of the C9orf72 gene. And it can be expressed hundreds to thousands of repeats long in the sense direction. But there's also an antisense RNA that's generated from an internal promoter in the gene. There are different ideas of how evidence of how it might cause disease. One way it might do that is that the RNAs can form G quadruplexes and bind RNA binding proteins in the nucleus and sequester these proteins away from what they normally would be doing, and thereby cause disease, sort of loss of function of these RBPs. Another way is that these RNAs can be exported into the cytoplasm and through non-AUG initiation by the ribosome, translate some of these repeats into short dipeptide repeats. And those may seed granules or lead to sequestration of RBPs in the cytoplasm. So one would imagine that one way to fix the disease is to target and deplete the sense or antisense RNA, which would solve both problems, whether it's an RBP sequestration problem or RNA translation problem. Unfortunately, we've seen some failures in this space where in ASO targeting the sense RNA seem to did not meet any secondary efficacy endpoints. And so one idea maybe is that you have to target the antisense RNA, or maybe both. And so we have scoured the literature and identified different zinc finger domain containing proteins that can bind the repeat and both sense and antisense. We've started engineering these and fusing them with endonucleases with the idea that you can then bind the repeats in the nucleus and then degrade them. And so some preliminary data can be seen here where we've used different zinc finger domains with a pin endonuclease from the SMUC6 endonuclease family. And you can see that when you co-express the G4C2 expansions in just 293 T cells with the some of the zinc finger domains, you can deplete the repeat. This can be quantified here with RNA dot blot imaging, right, of the RNA dot blots. And interestingly, if you express the C4G2 repeat, we also see some depletion of it with the zinc finger fusions. And that suggests that one reagent here can seem to bind and target both sense and antisense repeats. Now that was in 293 T cells, and so the ex-postdoc in my lab, now she has a faculty position in Vermont, Kathy had then started to differentiate these iPS cells, iPS lines from C9 patients. And the goal here was to get to a level of maturation that we can understand the impact of our zinc finger pin fusion on gene expression in different cell types, but also on obviously the repeat at the RNA level and the dipeptide proteins. So she grew this out for about 60 days, in which you can see mature astrocytes, interneurons, obviously motor neurons, and was able to then section them and then do RNA fish for the sense and antisense RNA. The sense RNA tends to be more numerous but smaller, and the antisense RNA tends to be larger and fewer, but you can see them. And when you treat these organoids with the zinc finger pin reagents, you can reduce the foci, sense and antisense in these spinal cord organoids. Also, this is true for the dipeptide repeats. Using antibody staining, you can see reduction of the poly-GA with the zinc finger pin expression. Now, the great thing about these organoids is that you can readily dissociate them, and you can identify different cell types from them. And then you can ask in the individual cell types, are you reversing the molecular signature of a C9-driven ALS? And we see partial or full rescue of these in astrocytes. Oh, I guess I mean astrocytes and other cell types within these organoids. Okay. The next stage would be to evaluate if these reagents can be delivered into the brains of mice, just to test for both safety as well as efficacy, maybe some reversal, if you're lucky, of some phenotypes, right? And so there is a mice model from Laura Rennan's lab that contains a bag of the human C9 expansion. You can see here in the C9 locus. And we worked with Stefan Paltz's lab, who studies ALS in these mice models. And his lab then did ICV injection of our initially GFP tagged zinc finger pin domains, packaged in AAV PHP EB. And this was at 19 weeks. And then we basically looked at the tissue at 28 weeks for expression and for other molecular phenotypes and cellular phenotypes. So you can see the AAV PHP EB is actually pretty well tolerated and distributes really well in the brains of these mice. Then we sliced the tissue and looked at poly-GA, poly-GP dipeptides, and you show reduction of these. By fluorescence, we can also see by dot plots and other analyses reduction of the repeats. I hear a pause. So did you want to ask a question, Chris, or no? Okay.
No.
Well, I think since you stopped there just for a moment, I thought that's very interesting. You have this model of ALS in an organoid composed of iPSC-derived neurons, and that was predictive that your Z1 pin, your zinc finger pin, would actually work. Would, I guess, reduce the amount of the aggregates that are forming. And then now you're showing that this is working in a mouse model too. I just found that interesting because there was this announcement from NIH just recently that, I'm sorry, it was the FDA. Just recently that
FDA,
the FDA announced that we should be really focusing more on iPS-derived models and non-animal models, more human-derived models. So maybe you could just comment on that just briefly.
Yeah. So the interesting thing is in ALS, at least, and probably other neurodegenerative diseases, the mice models or rodent models have not really recapitulated the disease phenotypes that you often see in patients. The other issue is that they're just different species. The genomes are different. So, for example, in ALS, you have typically a loss of TDP-43. It's another RBP in the nucleus, which may affect splicing of hundreds of substrates, right? And one of the critical substrates that are misspliced is this Stathman2 mRNA, or the cryptic exon in this mRNA. It's no longer repressed by TDP-43, and this exon is just completely missing in the mouse genome. And so there are many examples where you have features that are important in neurodevelopment, neurodegeneration that are just not found in the animal system, right? And so I think that it's very helpful though to leverage the animal systems for thinking about things like delivery and safety and toxicity. But for on-target biology, I think the human systems are cellular systems. If you can generate organoids or something like that, or iPS derived neurons, those are very powerful, right? As long as you can sort of start over time seeing, you can get the relevant biomarkers in there as well and see think those change. So I mean, I do think that we still, I would be nervous about dropping these things directly into people without going through a proper safety study in animals. But I think using a animal system to try to engineer that system to have the same disease phenotypes, I think that may be difficult in some conditions.
That's actually a really good distinction that you made, that the safety toxicity studies and animals, small animals, large animals, still are important in safety toxicity. But what you're saying is that for understanding whether or not your drug is going to work, or whether biological device, your intervention is going to work, having a human model is going to be better, superior, potentially.
Right. And I can actually articulate this here as well. So, for example, the C9 mice model in the original paper exhibits gross motor phenotypes, but these phenotypes are highly variable. And so it turns out that it's very difficult for us to leverage this particular mice model or other mice models to see if you get motor movement deficit. However, and I was just going to show you at the cellular level, you do see phenotypes. And so I'm just going to talk through this data and then sort of hammer home my point. So this is spinal cord sections from these mice models, the C9 mice model, and then compared to wild type. And you can see that these mice treated with different things. PBS is a control, pin only, so there's no zinc finger targeting, and then Z1 pin, and then same for the C9 mice model. You can see that at the cellular level, if you look at nu-N as a marker for healthy neurons, the C9 mice models are quite depleted for nu-N. They have more cleave caspase 3, so the cells are dying. You can see quite clearly here with the Western blot here. But then in the zinc finger treated C9 mice, you have restoration of the nu-N levels to wild type, and then same for a reduction of cleave caspase 3, right? So these mice models may not have overt motor phenotype, but we can still assess things like neuronal death and other maybe molecular phenotypes, and at least that's what we can do. And and so this is also true for cerebral cortex. So I just maybe hammer home the point. I think this mice model was still helpful for us because we think about how the AAV delivers, whether you can still see reduction of the negative phenotypes post disease onset, at least in the mouse, right? And so that's helpful for us to understand if you also get toxicity in other tissues, right? And then just to even push it even further, this is now using a wild type rat model that Martin Masala's lab, who's my neighbor upstairs, my building, is an expert on. And here he delivered as a safety, non-GLP sort of safety study, this zinc finger pin, now with AAV9, because AAV9 is what will be used in patients, not the AAV-PHPEB, right? And so AAV9 has been clinically approved in patients, so you're using the AAV9, and he's doing sub-PO delivery to create the retention of the vector within the spinal cord of these rats, and this would be the mode of transmission perhaps in patients. And he's performed a whole series of different studies, both grip studies, muscle fibrillation, looking at motor evoked potentials, and survival, past 5 months, and everything have looked terrific, right? So I'm just going to present a few slides here. So for example, here, maybe the videos are not working, but that's probably fine. Let's see that. Yeah, I don't know if it's working. But it just when looking at sort of open field motor performance, they all look the same. Maybe I can try to change my One second, I'm going to see whether I can get my mouse out of the way here. Nope, I guess the videos are not working. Fine. But also things like, do you see any issues with just motor evoke myogenic and H reflex responses? Because if you express these proteins in the spinal cord, maybe you see defects in interactions between the inner neurons, alpha motor neurons, and sort of overall electrical stimulated potential in the brain and spinal cord. And we don't see that, at least with this at this dose. So again, like, I'm not an expert in thinking about the non-GLP safety and tox, but these mice models are, this wild type mice is still helpful. And presumably we need to do this in non-human primates to be very comfortable with thinking about dosing and thinking about safety before you ever think about people. And so I just want to end with this slide. We are interested in this class of this RNA-targeted zinc finger proteins. We are working to try to understand their rules. Combined with our screens of which effector domains can do different things to splicing, stability, localization, blah, blah, blah. Perhaps we can combine them in interesting ways to then create novel platforms or modalities. And, because these are all human-derived, they don't have the same immunogenic concerns as bacteria-derived RNA modification systems. We spent many years trying to work with CRISPR RNA targeting for a long time. And I think that's still very difficult. They're neutralizing antibodies for these proteins. And they're very small. They are the smallest engineered protein that you can develop to bind RNA. And so we are excited about that possibility. Okay, I'll just stop here. I mentioned the Center for RNA Technologies. I also direct the Stem Cell Innovation Center. Thank you for your time and for the opportunity to tell you about science and our people. Yeah.
That was great, Gene, and we do have some questions. I think I'll let Dr. Brannon introduce some of the questions from the audience.
Sure. Yeah, so one here is, what are the potential benefits and risks of using AAV-based gene therapy like ZFP delivery in post-onset stages of neurodegenerative diseases? And I guess just a follow-up on that question would be, have you thought about different delivery modalities, like maybe as an mRNA payload or other forms of delivery for the ZFP modality that might be advantageous as compared to AAV?
Yeah, it's funny that AAV is getting, it's still getting a bit of a bad rep the last couple of years, right? But for deep brain delivery and for getting the payload to the right cell types, AAV is still superior, turns out, right? I think part of the issue with toxicity is people are worried about systemic delivery of AAV, and then you get accumulation in the liver or other tissues. I think for CNS diseases, if you can deliver this IT or by sub-PO, which is what Martin Masala has pioneered, I think that dramatically reduces the risk of leaking to peripheral tissues that may cause accumulation in toxicity. And so I think that helps. Of course, you can package the ZNPs, zinc finger proteins in other vehicles, except then you need continuous application, right? If it's a LNP mRNA, and LNP mRNAs haven't really shown clear efficacy from getting even through the blood-brain barrier, right? So then that is a different form of difficulty. One could argue you could deliver these in some sort of secreted way, but then you still have to get them into the cell, get them into the nucleus, get them to express the RNA. So I think for now this combination, like I always want to be very careful we don't sweep an entire modality under the rug because of specific concerns. I think this particular modality plus route of administration makes makes AAV like a really compelling product, right, in my mind. However, that also means zinc fingers may not work in other cases, right, where we want to say knock down a gene found expressed in the skin, maybe topically topical ASO may work, or siRNA may work even better. So just want to make sure that I clarify that. Yeah.
I guess I had a question too on the specificity of the Z1 targeting. So I guess how do you assess off targets, since the target seems it might not be that specific? And I guess what are the challenges that the ASO approach faced that you've been able to overcome, and what are the differences there?
Mm-hmm. Yeah, so I think what's interesting about targeting these repeat expansion diseases is that the substrate that you're trying to bind to and deplete is thousands of nucleotides long, pretty concentrated and reasonably highly expressed, right? And so the longer the target substrate, the more you are sequestering also the zinc finger protein or pin domain, such that if they're off targets, and we haven't seen, actually we've done RNA-seq on these and haven't seen dramatic numbers of off targets, or some, but they are at a different sort of ratio, right, from if you have a small target sequence. Here you have a large sink of a repetitive sequence. But still worried about that. That's why we did the safety study in the rat model. We were expressing the reagent in wild-type animals, and we haven't seen any safety issues, right? We don't know. Still, it's still rats, not NHPs, but that actually allays a lot of my initial concerns about off targets. And then your second question, Chris, if you don't mind repeating it.
Yeah, I guess I'm trying to remember what the main challenges that the ASO approach faced, and and how those were overcome here.
So the ASO approach is not targeting the repetitive sequence. It's targeting the sense strand message, right? And there's some indication that the antisense repeat actually increases when the sense repeat is depleted. It may be that the disease is driven primarily from the antisense, but I would be surprised if the sense also doesn't contribute to disease pathology, right? So ideally you have something that targets both. I think that it remains to be seen if one can deliver two different ASOs, because each ASO will have also their own fair bit of off targets and potential toxicity, right? So then you're multiplying both risk factors together. So I don't know. I'm optimistic because the ASOs have a proven route of administration for SMA, SOD1, maybe for C9 as well. But I don't know. So I think it helps to continue to develop different versions of this, right? And I think that the AAV approach at least is a one-time injection, even though it's a sub-P or IT, whereas the ASOs may have to be delivered every few weeks to every few months. So there's a different risk trade-off there, right? Ultimately, you can see from the case of spinal muscular atrophy that the patients leverage all the different modalities, small molecule, AAV, and ASO, because that may provide different ways of de-risking the disease at different stages of their progression.
There's a related question, Gene, from the folks back home about the pathophysiology. So you mentioned briefly that there's two potential mechanisms. The hexanucleotide repeat may be binding to RNA binding proteins and causing some problem there. The other possibility is that the hexanucleotide repeat is actually getting translated into protein that's causing the cytotoxicity. But with respect to the former, the question is, what is the mechanism if the RNA binding protein is the problem with this hexanucleotide repeat? What is going on there? What do you think is causing the pathology? Is it just providing a sink for RNA binding proteins and causing a deficit in some RNA binding protein, or is there something more?
Yeah, that is a model that the field has proposed. I think primarily based on studies of a different repeat in a different disease, myotonic dystrophy, where the CUG repeats in the DMPK gene and myotonic dystrophy type 1 sequesters this muscle-blind RNA binding protein, and the disease is primarily a loss of muscle-blind function that controls splicing. So I think a lot of the field has thought through that this may also be a cause, maybe causative, right? There's another one I didn't mention here actually, that this repeats is found in one allele of the patient typically, and the other allele is wild type. And the repeat themselves also cause a decrease in expression of the C9 gene itself. And there's some evidence that maybe the C9 gene itself is important for the disease pathogenesis. So if that's the case, then the ASO approach and our approach, which destroys the RNA from the disease allele, but not repairing the haploinsufficiency of the other allele now, may not fix it, right? And so that's something that we don't know as a community if that's true in patients, and we're evaluating.
Do you want to ask the next question?
Yeah, sure. So I'm kind of curious about expanding on this. So you have this Z1 domain, but what are you adding to the toolbox? Are you focusing on first zinc fingers as a class, and then getting all the rules down to maybe sort of make tandem sort of designer RBPs, and then will you be adding like different classes like PUF or other RNA binding domains to the toolbox?
Yeah, we have tried PUFs for a number of years. I think, Chris, you may know this, and the window length of binding for primary sequence is still relatively small, like 8 nucleotides. Maybe you can expand this. I think Ed Boyd and other groups have tried to expand this to 15. But we've had trouble with toxicity expressing these proteins to target RNA. So at least for us, we have stopped with the PUFs. We've also explored other modalities like using small nuclear RNAs, and so we have developed small nuclear RNAs. Aaron in the lab is developing them that can do A to I editing and pseudo-retylation of stop codons or premature stop codons. Those have legs. They're just they have different strengths and weaknesses to the zinc fingers. I mean, for me, I'm quite intrigued by both the basic biology of like what determines specificity for the zinc fingers. As a class of RBPs, they do a lot of different things, so that's quite interesting, and we're following that up. I think the other part is, since we're gathering data on what they might bind and how they bind, can we start assembling that into rules with AI and with large data production methods that we're familiar with? And so I think that may give us a toolkit. I don't know if it ultimately will be superior to other things, but I think they all play some role with some indications, right?
So that's really interesting, using zinc finger domain as a therapeutic modality. But I have a question just based on the conversation you guys had. What is a PUF? Can you describe that for us? Well, I don't know what that is.
Yeah. These PUF proteins belong to the familial response element binding protein family, PUM proteins, familials. And they were, I think, first discovered in yeast, I think, to bind unique sequences. They occupy a crescent-shaped conformation when binding to I think the backbone of RNA. I could be completely wrong, right? I don't remember now. But they can be engineered because they have the ability to be modular, and you can move the domains around to get you different specificities. I think they're very promising, but we have seen some toxicity with those as we express them in the CNS. Also, but perhaps other tissues. So I think people have to use them for myotonia dystrophy, and maybe in muscle that seems to be fine. So that's another potentially interesting class.
So for the zinc fingers, a lot of them have dual DNA RNA binding activity. Have you thought about leveraging that dual activity for anything? Maybe like bringing them to sites of transcription and then bringing an RNA binding domain along with it.
Yeah, that's a powerful idea, right? Because keep in mind, the folks are probably more familiar with the DNA-based zinc finger recognition, right? That companies like Sangamo have pioneered, and we've shown that some of the zinc fingers bind both, albeit it's a small set that but the zinc finger containing proteins bind both RNA and DNA, but presumably they have different zinc finger domains that bind DNA and some that bind RNA. One could imagine combining that so you get DNA specificity in terms of localization, chromatin interactions, or eRNA interactions, right? To bring it to affect transcription. Yeah, so that's all in the realm of possibilities. We just haven't had the haven't had the bandwidth to explore those. But those are interesting ideas. Yeah.
Well, Gene, we're coming to the top of our hour. I want to thank you for sharing
Great.
your ideas with us, and so you're doing some great work there at UCSD and also at Stanford, and bridging that gap between the basic science mechanisms of RNA biology and turning it into something that's useful for people. So congratulations on that, and thanks for sharing that with us today. Well, thank you for joining us
John,
again.
and Chris. Yeah, thank you so much, and good to see you.
Thanks all for joining us at the cutting edge of RNA therapeutics, and join us next month.